Hello, welcome to the office hour. Today we are going to do a demonstration of a technique that's commonly used in microbiology lab. That is the strict isolation. Are you ready? Let's go. Today I have with me several probiotic products like yogurt that I just grabbed from grocery store. All of them have supposedly live active culture. For example, here this is my favorite one here, Jobani yogurt. It has the several bacteria active live culture. It's written over there. I also have several other one here, including this Yakult, which I used to drink a lot when I was little kids, and several other one, and they all have supposedly active life culture. So what we're gonna do today is we are going to take sample a little bit from each of these product and then perform the 4 quadrant trick, which is the technique that you will learn in typical general microbiology lab, and see if we can isolate these microbes. Okay, so to begin with, I have with me several bacterial standard nutrient agar plate that you can find in microbiology lab, and the products here, probiotics over there, and I also have this perhaps the most one of arguably the most useful tool in microbiology lab, which is inoculating loop, and this is what we are going to use to perform the strict isolation. As usual, we have the Bunsen burner, and I think we are ready for it. Before you perform any laboratory activity, it is always a good practice to label your stuff. So here we are going to put probiotic products on the agar plate. So it is best to label them so we can identify which agar plate contain what sample, right? But before we talk about that, let me just show you. Here's the bacterial standard nutrient agar that you can find in microbiology lab. Typically, they are casted in this so-called Petri dish like this. You have the bottom where you have the agar over there, because this is the food for bacteria. Okay. Now, many bacteria will be able to grow in here, but I know some organism may require certain nutrients, then therefore may not be able to grow in here. So why do I say that? Well, I just want to let you know, in case there is nothing growing in it, doesn't mean that the company is lying and trying to trick you by putting the label live active culture and so on and so forth. It's not that, because sometimes some organism they require certain nutrients in order for them to grow. That is what we call fastidious. So if you are in the microbiology class, you probably learned the word fastidious organism, right? You remember that? Right. So here's again, this is the agar plate. You have the bottom that has the agar, and you have the lid right there, right? The lid, right? And when you label your plate, it is always best to label it at the bottom, right? Because if somebody come into the lab and they've switched around the lid, which hopefully not happening, but if that such thing happen, then at least your label stay with the agar where your organism will be found, right? So if you label on the lid put somewhere else, then you're screwed, right? So okay, so and here I have a stack of plate that I already pre-labeled. So to save time, generally when you label the plate, you want to label them on the side like this. See, here's on the bottom. Here is the label. You want to label them using the fine tip Sharpie, something like this, so that your label doesn't become a big blob, you know. So basically, you label it on the side. Make a legible, but you don't want to make it too big. And the reason why you want to label it on the side is because you don't want your label to obscure your growth later on, right? If you imagine if you put your label really big at the center, later on when your bacteria grow, you will have hard time looking at what is that, you know. So anyway, so that's basically how you label it. Now that we have all the plate labeled, so we are now ready to perform strict isolation. Now in the classroom, you may learn different version of strict isolation. Again, the purpose of strict isolation is for you to separate this microbe from each other so that they land separately. Individual cell land separately on the plate, and that individual cell later on will grow. They will divide from 1 to become 2, 2 to become 4, 4 to become 8, and so on and so forth until after 16 hours or so, after some period of time in the incubation, you see that this initially individual cell form a visible mass that you can see without microscope, and that visible mass is what we call colony. Okay, so how do we do that? Well, today I am going to simply perform one type of strict isolation, which is probably something you learn in microbiology lab, and that is what we call the 4-quadrant strict. As the name suggests, 4-quadrant, it has 4 quadrants. The first thing that we need to do is, of course, to get our Bunsen burner up. So here is the Bunsen burner. I'm going to turn on the gas. There you go. So now let's start with the loop. Now, when you perform strict isolation, you want to master your aseptic technique already, and this is why in typical microbiology lab, you learn about aseptic technique, and then once you master that, you perform strict isolation, which is a type of pure culture technique. Before you take your sample, here, you need to make sure that there are no other bacteria on your loop, because if there is, then you are introducing contamination. So how do you make sure that there is no other bacteria on your loop? Well, you want to sterilize them, and the most common sterilization technique that you do in microbiology lab is the Bunsen burner. So basically, you want to flame your loop until it's red hot. Now, when it's red hot, you know it's sterile. If you have any biological entity there, the moment it turns red, it become ashes essentially. Now, let's flame our loop. Now you see it, it's orange. Now this one is sterile. Okay, so this is hot. You don't want to touch it with your hand, and also you don't want to leave it on the table, because guess what? Your table, even though you disinfect it at the beginning of the class, it has things drop from the air, from everywhere. So it's never a good practice to leave the sterilized loop on the table. Okay, so now let's take our sample. So here I am going to take the yaw plate first. So here I have the label yaw plate over there. So here I'm going to take this. Let's see what we got. There, just take some sample from there, a little bit, but this is kind of too much actually, but we'll see. Now, in the 4-quadrant strict, you want to make your first quadrant by depositing your sample in your first quadrant. So notice the way I open it. This is what I call the clamshell method. When you open it, you try to open the clam away from your mouth, so this lid serves as a shield in case, you know, when you talk, especially me talking a lot right now to you, spit can fly off. So this is why we want to protect our agar by doing this opening clamshell method. Okay, so here I'm going to spread the sample in the first quadrant here. Okay. Now, one thing that can be quite difficult when you perform this for the very first time is that people oftentimes they gouge the agar, they basically slice the agar. You don't want to do that, okay? So now that's my first quadrant. I don't know if you can see that. Yes, right there, right? So that's the first quadrant. One of the way for you to get isolated colony is you want to lower the cell density that is in this yogurt to very, very minimum amount so that every single cell can land quite far apart on your agar, because what you want to do is you want each individual cell to become colony, to become an isolated colony away from each other. So that's what you want. That way you can take, for subsequent process, you can isolate from that isolated colony. You can take sample from that and perform an experiment. Now, in order to do so, well, we need to spread this quadrant, the sample from quadrant 1 to quadrant 2. Now, to make the second quadrant, you will need to use loop again, but you don't want to use the same one that you just have over here, because this loop still have the sample from the same source, and therefore they will have the same amount of cell density. Then you may not get a good isolation. So what you want to do is you want to sterilize the loop again by flaming them. There you go. Okay, now it's red hot. Okay, this is very hot. Okay, and it's sterile. Now it is very, very hot, and therefore if it's too hot for you, it's most likely too hot for your poor bacteria also. So you don't, and this is one of the common mistakes that I found over the years, that people just sterilize it and then using the hot loop they streak right away. That's not gonna happen. Okay, your bacteria will die and you end up with nothing growing on your agar. So don't do that. Okay, so instead you may want to wait a little bit. Actually, ideally you don't want to keep talking like me, because then you may accidentally have a spit projectile goes into your loop. Ideally you don't want to talk like me. You quiet. So you know what, let's just do another sterilization. Okay, here you go, I talk too much. Okay, so now you want to let it cool. One way to do that is either you can wait around, but sometimes it just seems the time fly by and it's a waste of time, right? So if you are impatient, then what you can do is you can try to touch your hot loop onto the area of agar that you believe there is nothing, you have not deposited any cell. Okay, just put it over there, and at first you will see the mark, because when your loop is hot, it will sort of melt the agar underneath and leave a mark, something like that. I don't know if you can see that. Yeah. So and then you do it multiple times to until you don't see any more mark. And when that happens, you know your loop is cool enough, and you can use the loop to drag some of your sample from quadrant 1 to quadrant 2. Okay, here we go. Alright, so we make our quadrant 2. We're gonna continue to quadrant 3, but don't forget to always sterilize your loop before making the next quadrant. Okay, here we go. Alright, we're done with quadrant 3. Now let's make our last quadrant. Okay, as usual, sterilize it. It's done. So let's make the last one. Here we go. Alright, we are done. So now before you place your tool away, you want to sterilize it one last time. Here we go. Alright, that is basically the strict isolation technique. So I am going to do the other 5. Once we are done with the strict isolation, we are going to put it into incubator and we are going to wait maybe about 16 to 24 hours next day essentially. Hopefully by tomorrow we get something growing on your agar. And yeah, I guess that's pretty much it. So I'll see you guys in the next office hour.